For you.
Yes, thank you very much, Isabelle, and Marilyn too, and all the organizers of the IFSCC. I'm really happy today. I'm pleased to share with you our work that I'm trying to share that we've done like for the 5 last years about our development of for a digital solution that allow us to use multi-omics technologies in order to better understand epidermis reconstruction and ultimately epidermis regeneration. So why, can you see? Yes, my screen is working. So why are we interested in epidermis regeneration in general? And you all agree that it is really an integral topic for cosmetics, because epidermis, it works as a shield that protects us from external aggressors, from, for example, UV and ozone exposure, and for example, from mechanical injury. And further, all the cosmetics that are applied on the skin, they go through the epidermis. So it is very interesting for us to understand how does epidermis work and how does regeneration works. And a better understanding of those biological mechanisms underlying epidermis regeneration will allow us to discover new biomarkers and ultimately to develop targeted cosmetic actives. So one of the best way to analyze epidermis regeneration and better understand all the biological mechanisms behind this process is using omics technologies, which have been like in progress since 15 years now, 15 to 20 years now. And the main, let's say the main challenge with those technologies is that they produce a huge amount of information. So we have like one major situation is how we are going to analyze those information, how are we going to transform it into intelligent knowledge, and how are we going to unlock all the knowledge behind those omics information. This is why today I'm going to be sharing with you our digital solution to transform all those multi-omics data into an intelligent knowledge to better understand the skin regeneration. If we go, broadly speaking, we can talk about 2 different ways of analyzing omics data. The first one, which I show here, if you follow my screen, it's called single omic analysis. And in this case, each of the different omics data sets, each one corresponding to a separate molecular layer, let's say genes, proteins, metabolites, and even genome, we can talk about a single omic analysis when each of those data sets is analyzed separately, and then the conclusions, they are compared and integrated together. However, as you all agree that inside an active cell, all the biomolecules, genes, proteins, and metabolites, they are in constant dynamics, which means that analyzing each of those single omic layers alone won't be enough to have like a complete picture of what's happening really inside the cell. And to answer this question, there are new ways of analyzing omics data that we call integrative multi-omics analysis, in which all the different layers are generated, and then there are kind of interconnections between the different biomolecules of each layer in order to integrate the different data sets. And in this way, we could analyze jointly all the data sets, and the conclusions will match all better, let's say, the biological mechanisms. However, with this second way of analysis that we call the integrative multi-omics analysis, we have like many challenges. First, that we need to generate and process the data sets simultaneously. We need also to develop adapted pipelines and analysis workflows that allows to really answer our needs. And third, we have also a big problem today with interpretation and transforming this data into real and scientifically pertinent knowledge. If we go through the literature today and we just check for epidermis reconstruction and multi-omics analysis, what we could see is that since 2009, there are like